Hi, dear students. A warm good morning to all of you. Welcome to one more session of immunology, and today we are going to talk about the complement system. Depicted here is an RBC whose membrane has been lysed by some agents. Now, if I ask you what is that, you will say it is a complement-induced lesion which is being depicted here because it is already written on this figure. But what is that? That's what we are going to explore. Before we go into the details of it, I would like to make you fresh with some terms like serum, plasma, and they're all. Now, what is the plasma and what is the serum? You take your whole blood and you just centrifuge it. The RBC gets settled down, the WBC and platelets would be above that, and there would be a fluid filled portion of the blood above, and that is referred to as the plasma. Now, the same blood, if you allow it to clot, you can see that the WBC platelets as well as the RBC will form a clot. They will get clotted, and they will be found at the bottom of this particular tube, and there would be a fluid portion remaining. That is referred to as the serum. So, serum is nothing but plasma minus the clotting factors. So, whatever, let it be serum as well as plasma, it does contains the antibodies which are being produced by the what? By the B lymphocytes. So now let's move on to another thing called the serum versus the antiserum. Both the term serum as well as antiserum do have the term serum, and you must understand that they both of them contain antibodies. But what is the difference between it? Serum is something which is normally being found within your body. That is, excluding the clotting factors, you will have, if you allow your blood to clot, the fluid portion which is coming out is referred to as the serum. So, the serum will contain all antibodies which normally you are getting exposed to. But in some cases, an animal or a human might be intentionally injected with a particular infectious agent or an antigen so that it develops antibodies against that. And the serum which is being developed in that human or the animal source on intentional exposure of an antigen is referred to as the antiserum. And this antiserum is usually being prepared when we need a high medical need or might of that particular antibody, or might be when we have to use that antibody for diagnostic purposes or anything like that. So I hope you got a concept of that, and now let's move on to the complement. In 1890s, Jules Bordet at the Institute of Pasteur in Paris, he took sheep antiserum. You know what is antiserum now? That is, the sheep was being injected with a particular antigen, which is that disease. The pathogen was Vibrio cholera. It was injected with Vibrio cholera, and the blood serum of that sheep was taken. So that is called the sheep antiserum. So the sheep antiserum to the bacteria Vibrio cholera was being taken, and it was viewed if that antiserum could lyse the bacteria, and he saw that it was able to lyse the bacteria. Now, what he did further is he heated the antiserum, and he checked for the bacteriolytic activity. That is, heated the antiserum and then mixed it with some Vibrio cholera. But he found that Vibrio cholera was not being able to be killed by this antiserum. And secondly, what he did, he took some normal syrup, that is, which was not being exposed to Vibrio cholera. He took that normal serum and mixed it with mm, what this heated antiserum, and he tried bacteriolytic activity. That time he saw that the heated antiserum, which had lost its bacterial activity, when it was mixed with a normal serum, it was able to kill the what Vibrio cholera. So, what was the reason behind this? Uh, See, he concluded that bacteriolytic activity of Vibrio cholera or against Vibrio cholera was being caused by two different substances, and one substance of that was being lost, where was being destroyed when the heating process was being done, and one substance was being retained even after, or it was resistant to heating process. And the um, and also you can see that 56 degrees centigrade at 30 minutes will inactivate a complement. And later on, Paul Eldridge in Berlin, he also carried out similar experiments, and he named the substance as complement, defining the activity of the serum that completes the action of antibody. And Bordet won the Nobel Prize in 1919, and that is known as a complement mediated bacteriolysis. So, let me just explain to you here. In fact, antibodies are resistant to the heating process, and they do not lose their what bactericidal activity, but complements are some proteins which will lose their activity their lighting activity on heating. So, uh, if complements are not there, the antibodies cannot work. When you heated the serum, the antiserum, the complements of that antiserum were being lost. But when you took a normal serum, which was not exposed to the antigen, but it already contained complements. Okay, so that when that normal fresh serum was being mixed with the antiserum specific for Vibrio cholera, and you can see that bacteriolytic activity was being obtained. So a concept was being developed that not only the antibodies, certain other substances are also needed for the completion of the action of antibodies. And these proteins, which are needed for the antibodies to lyse a bacteria, or which completes the action of the antibody, was being named as complement by Paul Eldridge. And the founder of the component complement, Jules Bordet, sorry, he was awarded the Nobel Prize in 1990 for this complement mediated bacteriolysis. Now, what are the roles of these complements? Complements, they play a major role in the humoral branch of immune system. You know what is humoral? That is antibody mediated immune system. And although the discovery of complement and the most early studies were linked to the activity of complement following antibody, a major role of this system is the recognition and the destruction of pathogens based on recognition of a pathogen associated molecular patterns, or the PAMs, rather than the antibody specificity. So it is, though it was being mainly for the antibody binding, that is, we were just uh, pardon. Okay, we had discovered these complement system usually associated with the antibody binding, but it plays an important role in the recognition of pathogen associated molecular patterns. Okay, that is specifically to identify the pathogens. Also, the complements play an important role. Now, what are the functions of these complement? Mainly, as I told you, it causes the lysis of cells and bacteria and viruses when they enter into the blood. The antibodies are mediated. And it also causes a process called opsonization. What do you mean by opsonization? It is to promote the phagocytosis of particulate antigens. Here, in the blood, you have the antibody mediated, and in the when it also promotes the phagocytosis of particulate antigens. Okay, you know the particulate antigens are those just the opposite of soluble antigens. Now, and the binding to specific complement receptors on the cells of the immune system triggers specific cell functions such as inflammation and the secretion of immunoregulatory molecules. So when where the binding in certain cells, there are receptors for complements. That is, there on the surface of the cell itself there are some receptors. You know, where the receptors are the places where something binds. So when the complements bind onto some cells, it would trigger the function of inflammation as well as the secretion of other immunoregulatory molecules, and also immune clearance, that is to remove the immune complexes from the circulation and deposit in the spleen and liver for to be further digested. That is being carried out by the or that is called the immune clearance. That function is also being carried out by the complement system. So we can see the lysis of the, this is a red color is the complement, If you have a particulate antigen, the complement will come and bind onto that, and it will cause the lysis of the target cell. And when an antigen antibody complex is being found, and when the complement goes and binds onto the phagocytic cells, okay, it has been a complement and the bacteria is there, and opsonization of that is being carried out, and sometimes it will go and bind onto some normal cells and causes the process of inflammation and all that. That is what is depicted here. And then on some cells it will go and bind and it causes degranulation. That is also a part of the inflammatory response. And once an antigen antibody complex has been formed, the complement also goes and binds onto it and causes the clearance of the immune complex. So the functions of the complement is of different types. Either it goes and bind to the particulate antigen so that what the lysis will happen, or it will go and bind to the antigen antibody complex and clear it, or in other cases it will bind with some bacterial components and that will be phagocytosed and opsonization can be carried out, or sometimes it will help in the processes such as inflammatory response, like on various WBC cells. So these are the functions of the complements, and usually this is another depiction, okay, of the same thing, of the various functions of the complement. And usually all these proteins are being formed, they will be these proteins will be formed in an inactive form, and you need to activate them within your body. And for that we have three different pathways called the classical pathway, alternative pathway, as well as the lectin pathway. And the in the classical pathway, it begins with the formation of the antigen antibody complex, and alternative pathway is initiated by the cell surface constituents that are foreign to the host, and it is antibody dependent. And the lectin pathway is activated by the binding of the mannose binding lectin to mannose residues on the glycoproteins or carbohydrates on the surface of the microorganisms, and it is antibody independent. So in the next session we would be learning about what are the different pathways of it and how are they getting activated. So let's stop for now. We will continue with the complement activation and the different pathways in the next session. Thank you for now.